Hello guys, and welcome back to another science video from the Howard. Today we're looking at enzymes, and in particularly how the pH of a solution affects the rate at which an enzyme breaks down starch, a complex carbohydrate. So I've got some varied pH buffer solutions here, ranging from pH 3 to pH 8. I've got some starch solution, and I've got some amylase here, which is my enzyme. Now what I need to do is take set proportions of each of these three solutions and get them to a temperature that I want to investigate. Now I'm going to keep that the same. That's going to be a control variable. So my temperature I'm looking for, I'm going for around about 30 degrees here. If it's cooler, it can take quite a long time for this practical to operate, so I'm going to work at about 30 degrees. I will record temperature, and then for the other solutions, when I go through the different pHs, I'll make sure I get to that same temperature as well to make sure it is a fair test. So I've got 3 test tubes over here, one for each of my solutions. What I'm going to do first of all is try and get my water bath ready for those test tubes to go in. Now I've got cold water in here at the moment, which is at just about 16 degrees centigrade. So I've got a pre-boiled kettle, and all I'm going to do is put some hot water in here, give it a stir, and then we'll try and get that to around 30 degrees centigrade. I'll wait for it to go up, add a little bit more in, until we get to 30 degrees. Now it's important when we actually come to measuring the temperature for our practical is that we actually measure the temperature of the solutions and not the water here. Otherwise our solutions may not be actually at 30 degrees. Okay, so we're at about 31 degrees in there, so I'm happy with that because it's going to cool down a little bit, so I'm going to put that to one side. Now I'm going to put my kettle away. And what I'm going to look at here is I'm going to put 1 centimetres cubed of our amylase in, of our enzyme. I'm going to use a syringe here, a 1 centimetre cube syringe. So I'm just going to pop that in and draw it up. I'm going to put that into the nearest test tube here. That's 1 centimetre cubed. Pop that down, and now I'm going to add 2 centimetres cubed of our starch solution. This is freshly made starch. It is important that we do get freshly made starch, as it does decompose over time. Now I'm actually going to do that again, because you can see I've actually got an air bubble in here, and I do not want to have an air bubble in there. It won't be an accurate measurement, so I'm going to just pull that out and pull it up again. Okay, we'll put that in our middle test tube. So, done with that, and I'm going to start at pH 3. Now, I'm just going to pour a little bit of our buffer solution into a beaker so it's a little bit easier for me to use a syringe. And I'm going to measure 1 centimetre cubed or 1 millilitre out of this. Okay, we'll put that into our file. Now the next thing I need to do, I need to put these into the water bath and just allow them to adjust to our temperature. So the first thing I need to do before I put them in, just in case I mix up, I'm just going to label them so it's nice and clear so I can use them again. So we've got A for our amylase, we've got S for our starch, and we've got B for our buffer. I'm just going to pop those into my water bath here. Now looking at my water bath is exactly at 31 degrees at the moment. So I'm going to allow these to adjust, but I am going to be making sure I measure the temperature of our solution and not the water, so it's accurate. The temperature of the solution is what we are looking at here. Okay, so now I've left that to adjust the temperature. I'm just going to check. Now I have wiped my thermometer down and put it into the starch solution, and we are at bang on 30, so I'm happy I can begin to start. Now what I've also put on the table now is I've put a laminated card with circles on that is stuck to the table, and I've put some drops of iodine in each of these circles. The iodine is going to be a test for starch. If starch is present, we're going to see a blue-black colour. Now once starch starts to digest, because of the enzyme, we're going to see it break down, and eventually that blue-black colour will stop as we now digested that starch into sugars. So we'll start with the blue-black colour and we'll finish with the orange of our iodine here. So that is our test for starch. What I will do with these three solutions is I'm going to mix them together. Once they're mixed together, the process of digestion of starch will begin. So I'll then start my stop clock and I'll take an initial reading at time 0, and then I'm going to continue to take a reading at every 10-second interval, which will go into each one of these circles here. So all I need to do now is just mix these all together. Now it's important I keep this solution in the water bath to maintain that 30-degree temperature. If I took it out, it would start to cool, and then that control variable would not be there. Again, you can see the colour there. So every 10 seconds, I'm going to take a couple of drops, make sure I expel what's in the pipette, and then again, we can see that lovely blue-black, almost purple colour there. Okay, I'm going to stop my stop clock there. So we can see the colour change going throughout these time intervals here. So I stopped the timer on there on 6 minutes and 50 was the last reading I took there. And we can see that the colour has stopped changing. The amount it changed obviously dependent on the time here, and you can see there were still slight colour changes throughout until I got to a point where the iodine did not change colour, indicating that the starch had been digested. Now I would now repeat this using pH 4, pH 5, pH 6, pH 7, pH 8, and then I would then be able to plot a graph to be able to see what our optimum pH is for digesting our starch. So just a couple of things to note before we do that. It is really important to reiterate that I keep my temperature of our solutions the same. So the next time I do this, I'll use the same volume of my amylase, my enzyme. I'll use the same volume of starch, the same volume of our pH buffer solution, and I'll keep the temperature that they are at the same. Let's move on and have a look at our graph. Right, so I've drawn my graph here, and as we can see on the y-axis we have time. This is our dependent variable. Remember guys, dependent variable is also going to go on your y-axis. On the x-axis, I've got my pH. This was my independent variable. This was the thing I was changing. Now, I've plotted my results here. I used pH 3, and you can see my time here of 410 seconds. So I've converted the minutes into seconds. Now, my other plots here, you can see pH 4 was the fastest time we had, and then the time increased at pH 5, pH 6, pH 7, pH 8. Now, I've plotted two lines of best fit here. Now, where they intersect, this could be my theoretical perfect pH in terms of the enzyme function, which would mean I'm looking at approximately just below pH 4.5. So around pH 4.4 here would be my optimum pH. This would break down the starch at the fastest rate, i.e. the smallest time period. Now my line of best fit here is not too bad. My results are all right. They're not too far away from each other. I could have perhaps tried to do this with more pHs, so I could have done pH 5.5, 6.5, 7.5, and that could have given me more accurate results. But I'm fairly comfortable here with knowing that my optimum pH is around about 4.5, just below 4.5. Now, This is bacterial amylase, and we know that should be around where its optimum pH is for digesting starch. We're not looking for pH 7 up here. We were expecting it to be around this point here.